Hello, everyone, and welcome to our webinar series titled To Immunity and Beyond, focusing on immunity and infection in donation and transplantation. My name is Erica, and I will be today's moderator. Firstly, I'd like to acknowledge that the CDTRP is hosted by the University of Alberta, which is located on Treaty... Shut up. Sorry about that. Located on Treaty 6 territory, a traditional gathering place for the diverse Indigenous peoples, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saluto, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to influence our vibrant communities. I encourage you to write your land acknowledgement in the chat box. We would also like to acknowledge CIHR, whose generous support enabled us to host these sessions. Today's session is recorded and posted on the CDTRP website and YouTube channel. During the presentation, please keep your mic off. If you have a question or a comment, you can raise your hand with the raise hand button or type it in the comment section. We are pleased to have Drs. Ian Roger and his trainee Ryan Wu present their research titled Induction of Tolerance of Bone Marrow Graft by Trogocytosis. Dr. Rogers is a University of Toronto professor and Lunenfeld-Tanenbaum Research Institute scientist specializing in stem cell research and regenerative medicine. His work developed 3D tissue cell systems and combines stem cells with decellularized organs to advance cell therapies. He is a scientific founder of the Inspect Life Bank and contributed to therapies for spinal cord injury and vascular diseases. Ryan Wu graduated this year from the University of Toronto with his majors in physiology and cell and molecular biology. As a fourth-year student in the Rogers Lab, he studied trogocytosis in human and mouse MSC co-cultures. This work inspired him to pursue a master's degree screening for trogocytosis enhancers and evaluating their potential in vivo transplant models. Speaking for myself and hopefully many others, I'm very excited about your presentation. I will now hand it over to you.
Thank you for having us here. So I'm Ryan. I'm a student in Dr. Ian Rogers' lab, and today we'll be presenting our presentation on the induction of tolerance to bone marrow transplant, bone marrow grafts by trogocytosis. So HLA mismatches are common in bone marrow transplants. A study in 2017 showed that 26% of Canadian bone marrow transplants involved at least one HLA mismatch. This is influenced by several factors such as patient ethnicity, the types of registry used, and the types of tissues that are being donated. So Caucasian patients are more likely to receive perfectly matched or closely matched bone marrow transplants when looking at both the Canadian and international registries. And this is because most bone marrow tissue donors are Caucasians. However, when looking at cord blood donations, this disparity is more equalized with similar percentages for both Caucasians and non-Caucasians. HLA mismatches increase the risk of transplant rejections as they cause donor or host cells to be recognized as foreign. This can manifest in two ways. The first is transplant rejection, where host immune cells attack donor cells. And the second is graft versus host disease, where immune cells in the donor tissue attack host cells. The mechanism for this is immune cells such as NK and macrophages can recognize the types of MHC class I expressed on different cells through receptors on the surface. So if a donor cell expresses the same type of MHC class I as the recipient, then that cell isn't lysed. However, if the donor expresses a different type of MHC class I, these NK cells or other immune cells can recognize it and then cause lysis of the donor cell.
Ryan, is this me?
Yep.
Okay, thanks. So I'm going to give you a bit more background. So trogocytosis is an interesting name. It means to nibble, and it involves the transfer of membrane sections and intact proteins between cells. And this sometimes seems to happen randomly, although it really doesn't. What triggers it is really cell signaling. So when there's two cells are signaling each other, sometimes what happens is chunks of membrane will be transferred from one cell to the other. So what's important to keep in mind is in vivo and in vitro models that we've done and other groups have done have shown that when you get a transfer of protein from cell A to cell B, this is just surface proteins. The nuclei don't actually fuse or transfer. So there's no genetic material, just protein. And this is kind of unique. And as you'll see in the models we're going to show in a minute, we've shown that mouse proteins will transfer to human cells and vice versa. So it's quite interesting. Next slide. So as I alluded to, the mechanism is actually called, it's an exchange of lipid rafts. So if we think about what cell membranes are made of, they're basically made of lipids with proteins floating around in them. Some are anchored, some aren't. But if you kind of think of the lipids as a raft with maybe anywhere from 1 to 10 proteins could be embedded in a section, that whole section could be lifted off and moved to another cell during cell signaling. And the other thing that's important to remember in this is that there's bystander molecules. So sometimes a certain receptor will be the trigger of trogocytosis, but if there are other proteins nearby, they will get carried along in the lipid raft. And that's why sometimes it's difficult to sort out what's actually going on with trogocytosis. In one of our models where we used human cells and NOD-SCID as a recipient, and also mouse cells and NOD-SCID mice as a recipient, we showed it can be bidirectional, so it can go both ways. But sometimes it, basically what happens is the cell develops, when it takes on new proteins, it develops kind of like a new phenotype. And there might be a selective pressure to maintain that cell because it now has new proteins. In some cases, it could be that the cell gets marked for destruction. So sometimes it looks like it's only transferred in one way, but that's generally a selection pressure, which we'll show you in a minute. Next slide. Okay, so rejection and tolerance is really complex. And it seems like all the cells of the immune system are involved. And last month's talk by Dr. Andras Nagy, he talked about this, and he has made, and he talked a little bit about a universal donor cell he made where they changed 8 different proteins on the surface as a way to protect the cell from macrophages, natural killer cells, B cells, and T cells, and other immune cells that are involved in graft rejection. So in order to kind of simplify our model so we could understand what's going on, we used 3 different models. So the first one was using human donor cells in a NOD-SCID mouse recipient. And when you read about NOD-SCID mice as a model, they are not supposed to reject tissue, but they kind of do. It's a little complex. And that's because they don't have T cells or B cells, but they still have residual natural killer cells and macrophages. And depends on how you work with the NOD-SCID mice, they remain inactive. So in some cases, you can transplant human cells and they won't be rejected because there's technically no active immune cells. But when you do a bone marrow transplant where the mice are irradiated to kill off their bone marrow so we could then put human cells in, that radiation actually activates the NK cells and macrophages. And we use this to our advantage because it allowed us to look at what happens with NK cells and macrophages only, no T cells or B cells. In the second model, we use NOD-SCID again, but this time we use mouse donor cells. So putting human cells in a NOD-SCID mouse, what's unique about this, and again, we could use this to our advantage, the human donor cells never produce a mature immune system. So the cells will be in mice for up to a year, but they'll never form a fully functional immune system. So they'll never wipe out the mouse recipient cells. So there's always going to be some of these mouse immune cells there. In the first model, they're going to constantly be targeting the human donor cells. And what we could see over long term is that trogocytosis is an ongoing process that protects these human donor cells over a long period of time from rejection. In the second model, where the new mouse donor cells are fully capable because the bone marrow environment is right, it's mouse into mouse, they can actually take over, form their own immune system, and then wipe out the non-skid mice. And we'll show you data from that. And in both those models, again, it allowed us to focus on natural killer cells and macrophages and their role in rejection and tolerance. In the last model, what we did is we try to mimic what would happen in a human-to-human transplant, bone marrow transplant. And in this case, we used a fully, like 2 mice that have different, there basically would be a mismatch for each other. So if you kind of think about in humans, the expectation is if we put the bone marrow from the BALB/c mice, they have a MHC, which is H2KD, and we put it in a recipient, which is a black 6 mouse, which has the MHC H2KB, so they're not a match. They're fully immune competent. We would expect the cells to be wiped out. And what this model allowed us to do is see if there's any tolerance, any protection from T cells, because these mice have functional T cells and they have functional B cells. But we only looked at what was happening with the T cells. So the principle behind using these 3 models is to break down the immune system so we can study 1 or 2 different immune cells at a time and try to deconstruct the complexity of what's happening in rejection. The other reason we're using bone marrow is because it's a lot easier to work with mice and do bone marrow transplant than it is to do a kidney transplant or a lung transplant. So we're really hoping that this model will translate to solid organ transplants. And I'll show you a bit of data that suggests that it does, that the bone marrow model is representative of what might happen in a lung, heart, or kidney transplant. Okay, so this is one of the first experiments we did. And there's a lot going on, but I'll try to keep it simple. So we want to, in this model, we put in 3 different types of human cells into a mouse. So we did a human bone marrow transplant into a non-skid mouse. And then we followed the human cells. And what we did is we used, first of all, stem cells, which we call lineage minus cells. So we isolated just stem cells, blood stem cells. And in the second group, we used lineage positive cells, which are mature cells, and they don't contain any stem cells. And this is actually an important control, which you'll see in a minute, because it's possible that when we see human cells with mouse proteins on them or mouse cells with human proteins on it, it could be from cell fusion. And if it's just cell fusion, the lineage minus cells would be positive in the short term, but not the long term. And I won't go into much more than that, but it's an important control. And then lastly, we just use total nucleated cells, which is where we combined all the cells together. We didn't separate them. And when you look on this, so on the y-axis, you can see we stained with H2KD, which is the mouse MHC. It's the mouse protein. And this is using flow cytometry. So every dot you see is a single cell. And then on the x-axis, because we also stain these cells with human HLA A, B, and C. So the H2KD is one color, and the HLA is a different color, green and red. So the lasers on the flow can separate the two. And what it allows us to do is look at what's happening with the different proteins. on the surface of these cells. So just when you look at this figure and you look at the top, you can see that we looked at day 7 bone marrow, day 9, day 12, 1 month, 2 months, 4 months, 5 months, 7 months. And then the last one is called 2 months human bone marrow. And the reason we did that is the other ones are actually all cord blood. Human cord blood, the stem cells are identical to the stem cells in bone marrow. So the cord blood was just easier to get. So if we look at, let's say, the 7 months, and it's the total nucleated cells, so down at the bottom. And if you look at the graph, it's a bit small and it's in quadrants. And if you look at the upper right quadrant, you can see that those cells are double positive for HLA-ABC, which is human protein, and they're also double positive for the mouse proteins. So if you kind of look at all the other graphs in the top right-hand quadrant, you can see even as early as day 7, we're starting to see cells that have undergone trogocytosis. And what's really important here, and this is where the induction of tolerance occurs, is the question we needed to ask, and the answer is yes. So I'll give you the answer and then I'll tell you the question is, are these the human cells that have somehow taken up onto their surface most H2KD? And if that's true, they'll be double positive, which we see, but they'll also now look like a mouse cell. So the way macrophages and natural killer cells work is even though they still contain like a human protein, which most recipient would consider as foreign and want to kill it, they also now contain the mouse surface proteins, which actually overrides the kill signal and keeps these cells alive. And that's exactly what happens. And we can see this is maintained long-term. Okay, next. Okay, so when we went to publish this paper, the obvious question the reviewers asked was, how do we know it's not just fusion and you have a mouse nuclei in there now and you have a human nuclei and they're both making mouse and human proteins? So the way we did this is we looked at chromosomes. And if you look at the first row, you'll see blue, and that whole column is blue, and that's DAPI to tell us where the nuclei are. In the top row, in the second picture, and then if you look down the column, you'll see 3 pictures with green, and those are human chromosomes, so the probe is green. And then if you look at the last picture in the top row and you look down, you'll see red for mouse. So what we did is we took our HLA human positive and our H2KD mouse positive cells. So these are cells that have both mouse and human proteins. And when we analyzed the chromosomes, we only got human chromosomes. So that told us this is a human cell which is the donor cell, and it took up somehow the mouse proteins, and it now has a mouse identity, and the mouse immune system is tolerant to it and wouldn't kill it. And all the other pictures are controls. So I'll just leave it at that. Next. Okay, now I'm going to try to keep this simple. So we wanted, as I said before, we wanted to see if this trogocytosis triggered protection tolerance, if it worked with solid organs. So the obvious thing to do would be to take a black 6 mouse and a non-skid mouse, for example, like we did with the bone marrow, and to take a kidney from one and put it in the other and analyze the kidney, for example, just like we did the blood. But doing kidney or any organ transplant in mouse is extremely difficult. There are people who do it. They're amazing technically to be able to do that, but in my lab at the time, I didn't have anybody who could actually do that. So we thought of a more complex type of experiment. And this was to make a chimeric mouse. So this is basically each of these mice, they have 4 parents. So there's a male and female black mouse and a male and female white mouse. They each produce embryos, and we kind of push the 2 embryos together to make a truly chimeric animal that has the genes of 4 parents. And then you can see from the coat color that if they're truly chimeric, they'll have some black and some white. So when we did this, we ended up with 3 mice. And what's interesting with this is part of their cells contain H2KB from the black 6 component, and part contain H2KD from the BALB/C component. And the reason that they don't attack each other is the same reason that our immune system doesn't attack our own cells, except for an autoimmune disease. It's basically our immune system is educated. So because this togetherness, this merge occurs during 8-cell stage very early on in embryo development, they won't attack each other. So the importance of this is it would give us a baseline to see if there's just naturally occurring trogocytosis. As I mentioned before, it is triggered by cell-cell interactions, normal cell signaling, and we wanted to see if bystander molecules, trogocytosis in general, occurs at a low level, let's say 1% of the cells, or a much higher level. The other thing I'm going to point out here is the black 6 mice also contain an enhanced green fluorescent protein. So this allows us to know which ones are the black 6 cells. And the reason that's important is we know the black 6 also have H2KB, but if it's transferred to the BALB/C cell, we wouldn't know that it was transferred if we didn't have a second marker. Okay, so hopefully you followed me on that. Okay, next. Okay, so we took those 3 chimeric offspring that we know from their coat color, they're chimeras. And this time we did flow. And what we did is we looked at green fluorescent protein on the x-axis, and we looked at H2KD in this color and PE marked for the BALB/C mice. And again, when you look at mouse number 1 in the top right-hand quadrant, 16.4% of their blood is from the black 6, the BALB/C mouse. Yes, and yes, yeah, okay, it's double positive. Sorry, I don't want to get too confused. So they're double positive. There is trogocytosis, and if you look at the 32% marker at the bottom, sorry, that's what I was getting at. Those are the GFP, which is from the black 6, and if they also contain the BALB/C, the H2KD, it'll be in the top right-hand quadrant. So that tells us that 16% of the time, the BALB/C marker H2KD transferred to the black 6 mouse. And then in the other animals, it's 13 and 15%. So this actually surprised us because this tells us that trogocytosis occurs in healthy animals where they're not necessarily being challenged immunologically, but this is quite a high level that occurs, and we don't really know why. Okay, so now let's go to the important, and Ryan, put up the, yeah. So this now, and it's taken me a few slides to get to this, but I wanted you to understand how we did this, is we could then take all these different organs, so brain, intestine, lung, heart, kidney, muscle, liver, pancreas, spinal cord, and testes, and we washed out the blood and we isolated the cells of the organ, individual cells, and we ran them through flow. We stained them exactly the same, and we looked at the upper right-hand quadrant, and we wanted to see if there was any trogocytosis. So in this case, did any of the black 6 GFP cells in the brain, okay, also somehow get BALB/c MHC on their surface, okay, which you wouldn't expect. And we can see that in the brain on the far right-hand column, 2% of brain cells actually somehow took on the BALB/c phenotype. So they had a mix. In the intestine, it's 3.5. The lung is 21%. So 21% of lung cells contain both, so on and so forth. And the only thing that the two things we learned from this is there is naturally occurring trogocytosis that goes on. MHC is transferred naturally, okay? We don't really know why. And it seems that if it's an organ that has a really high blood supply, that the trogocytosis seems to be a little higher. So this kind of jives with the bone marrow transplant where blood is involved. Okay, so the take-home message in all in this, and then I'll pass this on to Ryan, who will hopefully be less confusing than I just was, is that it looks like solid organs have the potential to develop tolerance through trogocytosis. And I'll leave it there.
All right. So in our next model, we have a mouse donor into a non-skid recipient. And in this transplant, we expect full eradication of the recipient's blood cells. We expect full chimerism as well if the donor cells engraft into the recipient. And we also expect graft versus host disease because the donor cells will form mature immune cells, which will then attack the recipient. So on the x-axis is the donor H2KB, and on the y-axis is the host H2KD. Similar, this upper right-hand quadrant, quadrant 2, shows all the trogocytosis positive cells. So when we do the transplant early on, post-transplant, we see that trogocytosis is very high, and it peaks at 74% 2 weeks post-transplant. However, as time passes on, what you'll notice is that the percentage of trogocytosis positive cells substantially decreases to about 15% at 5 weeks. This is because once the donor cells engraft, they'll form the fully functional immune system, and they won't reject donor cells. And so what happens is that you no longer have a survival advantage for trogocytosis positive donor cells. What that means is that you can get the survival of trogocytosis negative donor cells. And this graph just summarizes that. Where this contrasts with our first model is that with the human cells, once you transplanted those back in, you saw very high levels of trogocytosis like several months post-transplant. And that is because in that case, there was a survival advantage for those cells because the human cells don't form an immune system. When we look at an image of what a trogocytosis positive cell is, so here, this blue circle is DAPI staining to just show the nucleus. Red signifies H2KD, and green signifies H2KB. And so we have a cell that has both the H2KD and the H2KB. To confirm that this is not due to cell fusion, what we've done is we've taken primers that are specific for H2KD and H2KB and run PCR on them. And so they'll only show a band if that gene is present in the cell. So this is our first control, which is the black 6 donor. And as you can see, only the H2KB primer shows a band. On the other hand, the non-skid host only shows an H2KD band, which is expected. Now, once we take this trogocytosis positive donor cell and we run PCR, we see that only the H2KB primer shows a band. And that just demonstrates that what we're seeing in the cell over here is not due to cell fusion. And in our last in vitro model, this is to use fully immune competent donor and recipients. And so the donors are about C, which have H2KD, and the recipients are at black 6 with H2KB. And the reason why is to see if trogocytosis provides protection against T cells. So after doing a transplant, we use imaging flow to visualize the cells at 90 and 186 hours post-transplant. So to orientate you, this is the bright field, and in this column, this is what the cell actually looks like under the microscope. In this column called AF647, that shows all the H2KB protein on the cell surface, and this column labeled FITC just shows all the H2KD protein on a cell surface. So these are recipient cells, and what we see is that they acquire little patches of H2KD from the donor. Similarly, if we look at donor cells, what we see is that they acquire little patches of H2KB from the recipient. And when looking post-transplant over time, what we see is that gradually the percentage of trogocytosis positive cells increases over time. Initially from 0 to 90 hours, this is mainly due to an increase of trogocytosis positive recipient cells. However, as you look at from 90 to 186 hours, the percentage of trogocytosis positive donor cells increase. Now, moving away from in vivo models, we're now interested in vitro models. And the advantage is that rather than looking at the mechanism of trogocytosis, we can also test for ways to modulate trogocytosis. So the advantage is we can screen for candidate drugs that modulate trogocytosis, and these have potential clinical applications. Our goal is to see if we can apply a trogocytosis like modulator to reduce transplant rejection. The other advantage is that we can also test different cell pairs in a more controlled environment. So the model that we're currently using is human and mouse MSC co-cultures. MSCs stand for mesenchymal stromal cells. They're found throughout the body in different types of tissues and organs, such as bone marrow. And the other model that we're looking at is human blood cells with mouse MSC co-cultures, and this more closely models an in vivo bone marrow transplant model. So when I co-culture the MSCs, what I find is that MHC class 1 is transferred between both cells. So on the x-axis, this is H2KD, which is the mouse MHC class 1. On the y-axis is HLA, and this is the human MHC class 1. And what we see is that about 13% of cells are trogocytosis positive, and both the human and mouse MSCs acquire exogenous MHC class 1. This can be enhanced by adding interferon gamma into the co-culture, and what happens is that now the trogocytosis rate increases to about 38%. So interferon gamma is an inflammatory cytokine that's released when the immune system is activated, and it promotes several immune responses such as increased macrophage activity and increased expression of MHC class 1 by different cell types. So in this graph, what we can see is that more cells are trogocytosis positive, and some individual cells are also gaining more MHC class 1 just due to the shifts in the intensities in either direction. When doing imaging flow, we again see that trogocytosis positive cells acquire little patches of donor membrane and MHC class 1. So these are human MSCs. This again is the bright field, which is how the cell actually looks like under the microscope. And what you can see here is they have little patches of mouse MHC class 1. Similar, when we're looking at mouse MSCs, what we can see is that they acquire little patches of HLA. So we can also use a system to test for ways to inhibit trogocytosis. So normally it's about 13% when nothing is added. However, if you add BFG and heparin, what you'll notice is that the rate drops to 1%. Similarly, if you add blebbistatin to the culture, the trogocytosis rate decreases to about 6%. So both BFG and blebbistatin suppress cell contractility, and that suggests that MSC trogocytosis relies on a cytoskeleton-based mechanism. So why is this all important? The main application that we're interested in is reducing transplant rejection. So what you could do is you could isolate donor bone marrow cells, and as well as the recipient's MSCs, you could co-culture these cells together with trogocytosis enhancers, and then transplant the trogocytosis positive donor cells back into the recipient. And because these donor cells will now have the recipient's MHC or HLA, they kind of gain the recipient's identity, and they're less likely to be rejected. So in summary, the transfer of recipient MHC class 1 to donor cells by trogocytosis provides protection from NK and macrophage-mediated lysis. Trogocytosis can be modeled in vitro and used to screen for enhancers and inhibitors, and these enhancers could be used to reduce transplant rejection. Thank you.
Thank you for that excellent presentation. That was very insightful. I'm going to now open the floor to any questions if people have it. You can either raise your hand, ask it directly, or put it in the chat, how you feel comfortable. I'm going to start with a question. You mentioned very early on in your presentation that trogocytosis is kind of like a little nibble. What's the difference between trogocytosis and phagocytosis?
Good question. Ryan, do you want to answer that?
Yeah, so in trogocytosis, what's an important thing is that the donor and the recipient cells, they aren't lysed when trogocytosis is finished. Like they'll both be intact. Versus when you look at phagocytosis, what will actually happen is that one of the cells will be engulfed by other, and that cell is ultimately destroyed. And there is some literature perhaps suggesting that they might use different mechanisms. Yeah.
Okay, so it's the main thing is in phagocytosis, the cell is pretty much engulfed and dies. And yours
Yes.
is just a little nibble, and that will transfer some material.
Yes.
Thank you. I would then like to go back to slide 12.
Sure.
With the organs.
Yeah.
Yeah, there. So this data shows that you have you found that you had tissue-specific differences in the trogocytosis rate, so difference between lungs and livers. What mechanism do you think drives these organ-specific differences?
That's a really good question. And the short answer is I don't know. But right now, my hypothesis is blood flow, because there seems to be a correlation. But I don't know if it's great enough to really show that.
Okay.
Yeah. So just to kind of add to that, and to be honest, this is a super confusing area. When we started this, there was only 6 journals on Medline, 6 papers on Medline that talked about trogocytosis. Now there's thousands. And it's mainly been identified occurring like cell-cell signaling in the immune system, but it also occurs in other systems. So, for example, trogocytosis is used by cancer cells that kind of bump up against stromal cells in a body and will take some of the surface proteins, and that way they're protected against the immune system. So cancer cells have a way to hide from the immune system, and they use trogocytosis for that. So it's involved more, but we do see it in here.
We have a point in the chat, so it's from Francis. He's saying, fascinating mechanism. In your opinion, could this protein transfer via trogocytosis act as a driver for tumor development in the long term, much like what happens when trigomillus treatment over time?
Yeah, so yes, I think so. And again, as I mentioned, people do see that trogocytosis is involved in transferring proteins to cancer cells to help them hide from the patient's immune system.
I think just to add to that, CAR T therapy, I'm fairly sure that everyone's familiar with it, is there's a lot of interest in it. And one of the challenges with CAR T therapy is that what you often see is it'll work for a while, and then you can get like a resurgence of the tumor. And that the reason why is because of trogocytosis between CAR T cells and the tumor cells. And that kind of transfer enables tumor cells to not only hide from different CAR T cells, but it also can make CAR T cells less effective. And so, yes, just to go back to the point, there is a connection between trogocytosis of different cells and tumor cells and tumor development, and that's also why trogocytosis inhibitors could be very interesting clinically.
So I'll answer the other question in the chat. So the question is sorry, do you know if after trogocytosis, the recipient cells are able to produce by themselves the new MHC H2KD? So that's a really good question. So when we saw like at 7 and 9 months out in the human donor cells in a mouse, that's exactly what we thought was happening. But when we did the karyotyping and the fusion experiments and showed there's no DNA transfer, then we were kind of stuck because there's no gene transfer. So there's no way that a human cell can keep producing mouse protein because the gene actually isn't there. So then we had to figure out what's going on. And that's when we realized in the human model, there's constant selective pressure for cells to survive only if this transfer occurs of membrane proteins, and that therefore it must be going on constantly. So we don't know, we know we did some experiments just to look at turnover. So the MHC turns over about 24, 48 hours. So basically a trogocytosis event has to occur every 24 to 48 hours. So you have a population of cells that are always have this recipient identity. So no, they don't make it themselves. It's constant trogocytosis. It's constant trogocytosis.
Perfect. Can I ask you to go to slide 21, please?
Sorry, is this? Oh,
It's the IFN one.
Yes.
There you go. So here you observed that there was an IFN increase in trogocytosis in the MS co-cultures. Did you test for any other cytokines or inflammatory conditions?
No, we haven't. So one of the suggestions that I've gotten is to test for additional like cytokines as well, and different also types of interferons, because interferon gamma is only one type of a class 1 interferon. I won't dive into it, but yes, there could be other like inflammatory cytokines that are involved in this process, and they could potentially enhance it as well.
All right. And I have one final question. If anybody has any questions, put them in. If not, I think we will have my final question is, so what are the next step in developing trogocytosis-based therapies for clinical practice?
So I'll comment first, and then I'll let Ryan comment. So for me, I want to point out that despite what we see, everything tells us that bone marrow transplants shouldn't be rejected, but they still are. So for me, the question is, why do you have these protected cells, but ultimately it's the graft is rejected? And then kind of counter that against the literature where people have done transplant models in mice. And even they see this rarely in humans, but it does occur where like a kidney transplant will become tolerant. So a patient could be on immune suppression for a long time and then eventually get weaned off and the organ's not rejected. So, one of the questions I ask is that just ongoing trogocytosis and eventually enough of the organ cells are covered that it becomes tolerant, and therefore, if we can enhance it or push it to occur faster, is that one way to induce tolerance? And I'll let Ryan talk about his specific goals as a master's PhD student.
So I think so. This is the in vitro model, and I think the next step or the most immediate step would be to look at an in vivo model. So like another mouse to mouse bone marrow transplant model, and applying this these enhancers to test if they work clinically. And then I would say we might also be interested in taking a look to seeing if this could potentially apply to like an actual like human-based transplant. So kind of it's a lot of little steps to as a proof of concept to show that what we see in vitro with these co-cultures actually can be applied in vivo. And then I think kind of a little bit away from this is that it's also important to look kind of clinically to see if a clinical procedure can be developed to apply it. But yeah,
So good, big next steps, I see.
yes.
There we go. Okay, so Dr. Rogers and Ryan, thank you on behalf of CDTRP for presenting today. If anybody has any additional questions or comments or a suggested topic or speaker, please contact me at ekcroft@cdtrp.ca. I want to thank everybody for joining, and we hope we see you all for our next presentation on June 26th. We will be having Dr. Anna Konvalinka present her research on predicting kidney allograft outcomes and potential treatments using protein signatures. Enjoy the rest of your day. Take care.
Thank you.
Thank you.